images. And that's before augmentation. And by augmentation, I mean, while we're training, we are randomly flipping and rotating and scaling these images in order to better help our CNN generalize. And then for evaluation, we performed evaluation on 6 different HuBMAP data sets from the University of Florida, again, from lymph node, spleen, and thymus, each of which have either 19 or 29 markers. And 3 of them are on the HuBMAP portal at the moment, and the rest should be soon. And in order to evaluate the performance of the CNN classifier, we did cross-validation on all of the data that the CNN was trained on. Each of the faint lines is a different cross-validation fold for the ROC and PRC curves. And the purple line is the mean curve across all of the folds. And so we have on the left an AUC of 0.79, on the right an AUC of 0.66. So this at least shows that our CNN is learning the difference between a membrane protein and a non-membrane protein. We also wanted to look at whether the discrete wavelet transform actually improves the performance of the Ramsey's classifier. And we did this analysis only on the Goltsev et al. Data set, which is why the numbers look a little bit different. But with the discrete wavelet transform, that model does perform significantly better than without the discrete wavelet transform. So the blue is with it and orange is without. We also compared the performance of our membrane prediction model to other membrane prediction methods. The first with these 3 methods, so SURFI, PrediSci, and Signal P5 are all sequence-based methods for predicting membrane proteins. They actually predict signaling proteins, which tend to be membrane proteins. And these sequence-based methods take in as input the amino acid sequence of a protein, and then similar to Ramsey's, outputs a score between 0 and 1. And those correspond to the red, green, and orange curves on this ROC curve, and ours is the blue. And unsurprisingly, our method performs better. And I say unsurprisingly, it's because our method actually looks at the data itself, looks at the images, and actually has a context-dependent way of determining whether or not a protein labels membranes, whereas these just take in the amino acid sequence. Another method that we compared to is one based on Spearman's correlation, which appears in a paper in cytometry in 2015, which they're also looking at how to choose membrane markers for cell segmentation. And what they do here is that they take all the possible combination of pairs of proteins and then computes the Spearman's correlation coefficient for all of those pairs over the data set, take the top 10 most correlated pairs and then looks at the most frequent proteins present in those top 10 pairs and then selects that one. And since that doesn't give a score between 0 and 1, so, for comparison, we chose the most frequent proteins in those top 10 pairs and then normalized them between 0 and 1, where 1 would be the most frequent protein in those pairs and 0 would be all the proteins that are not present in the top 10 most correlated pairs. And that result is in purple, and that actually performs quite poorly compared to Ramsey's. And as a visual confirmation that our segmentation, the resulting segmentation seems reasonable, I've shown here, these are 2 images from the lymph node. The red here shows the Ramsey's output, so our weighted membrane channel. The green is the cytokine segmentation using that Ramsey's output. The blue is just the naive nucleus radius extension method, and then gray is the DAPI stain. And we can see in these images that the green, which is the result of our output, follows these membrane markers a lot more closely than, say, the blue would be, since the blue is just taking a simple radius out of the nucleus segmentation. Also, to further verify that our results, that our segmentation produces biologically meaningful results and segmentations, we obtained the cell by protein abundance matrix as from the cell segmentations and do a HuBMAP decomposition and then perform clustering on it as we show here. And then each each cluster is then labeled based on which proteins are most present in that specific cluster. So this is for the thymus. We've labeled these different cell types for the clusters we've obtained. And for example, in this blue epithelial cells, we labeled those based on the higher presence of pan-cytokeratin in these particular cells, where purple is going to be a little presence of this protein, and then green and then yellow is higher presence of the protein. Another example for regulatory T cells, we chose that label based on the increased abundance of CD4 and FOXP3 in these cells, specifically up here. We can also map back these labels to the original image just by coloring each cell based on the labels that we've given them. And so here we can see some nice spatial structures in the thymus. Specifically, this teal green shows double positive developing T cells distributed throughout, and which then interact with these blue cells, which are these epithelial and antigen presenting cells, which then drive their selection as single positive T cells, which are the red-orange colors here. And so to conclude, accurate cell segmentation is essential for analyzing codex datasets or spatial proteomics datasets. Determining the ideal protein markers for cell segmentation, however, is not always trivial. Ramses can choose these optimal protein markers for cell segmentation without prior knowledge, which is great. However, a caveat is that currently this HuBMAP segmentation pipeline is still dependent on the quality of the nuclei segmentation, because the cell segmentation algorithm is based on that. So if we miss some cells due to the errors of the nuclei segmentation, that won't necessarily be accounted for by the improved method that Ramsey provides. And yeah, I would like to acknowledge my lab, as well as Dr. Rossifal and Dr. Brusca from the University of Florida Tissue Mapping Center. Thank you.
Thank you, Monica. There are a couple questions in the chat, just 2 of them. How do you know that the combination is better than any of the individual masks?
Yeah, so I unfortunately don't have any images here, but we do have comparisons between just choosing the top ranked protein and using the combination. And we do have results that show that the combination at least visually looks better than just the top one mask, yeah.
And then someone asked if the membrane prediction software is confusing external plasma membrane with intracellular membranes.
Very possibly. So the membrane prediction software, we chose those just because those were kind of the only ones we could find that could predicted membranes, but they don't do it in a context specific way. They don't take as input like, oh, this is the tissue we're looking at, or this is the these are the markers we're using. So those methods don't really have any other context.
And then someone asked, is there any codex approach where you can get some absolute quantification information?
I am not 100% sure exactly what that person is asking. If
maybe I can try to explain it. This is
yeah.
So I guess that the information that you get with antibodies is you just see the CY3, but you don't know how that relates to actual quantity, number of molecules or concentration that you have in that cell. Is that somebody has done maybe some work where you put some internal standards or image next to it that you can say, this is the equivalent, this image in these cells, we count, I don't know, 1,000 molecules of this specific receptor or something like that. And then I had the second question that was, well, let's answer if you know, maybe if you can address that question. Did I explain it better?
Yeah, so to be honest, I am not quite sure. I just know that the images that I'm working with, like each of the pixels will have an intensity value. And so with the higher intensity value will be a higher at least relative protein abundance in that area. I'm not really sure about an absolute quantification of molecules or anything like that.
I guess that would be maybe I'm not sure if you know that either, but even in the when you use different CY3 versus CY5, even that I think there are some differences in the way that they perform those fluorophore, and even for the same antibody, you get different type of performance. So I'm not sure if anybody tried to tackle this issue of really getting to the point of even how good is the relative quantification or not.
Yeah, I am unsure if there has been any work done on that. But yeah, if anyone else,
thank you.
Awesome.
Anyway.
Thanks, Monica. There's a few more questions in the chat, and we're out of time for this section. But if Monica, if you want to check in the chat and see some of those other questions, you can feel free to jump in and answer those. And thanks everyone for dropping in those great questions and for that discussion. So we will go on to our next presenter, which is Andrea from NIH. So if you would like to jump in and get your screen share up, we can get started with the next presentation.
Thanks. Thank you guys so much. I don't know, I've got two computers because one of them failed. So I'm running off of two computers. So I might just turn my video off for the one. So we'll do that. And then I'll turn it on for the questions at the end. But thank you so much for organizing this talk. And I wanted to demonstrate first. So is this not? Oh, here we go, down here. So this is actually an image that I captured using the IBEX method that I'll describe today and that we developed in the Germain laboratory. And I wanted to put it in the context of a stained glass window. And so I relied on the Photoshop wizardry of my dear friend, Stefan Uderhardt. So I just wanted to share that with you guys. And indeed, I'm at the NIH. So, this cartoon, many of you guys are familiar with, of course, because it comes from the consortium. And I just use it to outline areas of impact and some of the things that I've really worked on, these optimal tissue preservation workflow and multiplex imaging in the Germain lab. And so first starting with tissue collection, which is important, obvious. And so this is a cartoon taken from a paper that's, under revision here, where it expands upon some of the workflows we developed in the laboratory to have consistent tissue handling, orientation, and so on. And this is a fixed frozen method and employs a milder level of fixative. And then we put it in sucrose for cryopreservation. And it has many advantages, including, and you'll see that it preserves tissue architecture and cellular morphology. It's also a milder fixative so that it obviates the need for antigen retrieval, and it's compatible with over 250 and more commercial antibodies. And importantly, as you'll see, I'll highlight specifically, it reduces autofluorescence from common sources, including red blood cells, tissue lipids. And then finally, what's fun and an interest in the Germain lab and others is because now we have this method that's archivable. We put it in -80 instead of room temperature for FFPE, but we can indeed then use it for multiplex imaging of greater volumes, which we typically employ. And so this is 400 microns, allowing you to see, the architecture of the tumor microenvironment in the liver. And so then, as opposed to, as we elaborate on analyses, so I'll talk to you about a multiplex imaging method we developed. And so, Monica did a fantastic job talking about the protein-based spatial analyses, such as Codex, and I'll just go quickly through that. This is a cartoon created by John Hickey in a recent perspective that we're writing with the Affinity Reagent Imaging and Validation Working Group. Thanks to everyone who's been involved. This is not an exhaustive author list, but I just use this to demonstrate that, there's this method, we're going after protein using antibodies. There's many different approaches using fluorophore labeled antibodies, oligo labeled antibodies, and metal conjugated antibodies. And so many of the technology developers are right here, part of the consortium. So we won't go into that great deal, just demonstrating or highlighting where IBEX fits in. And now I'll get into the method. So it is based on antibodies, fluorescently conjugated antibodies. It's a cyclic imaging method. And so, it's all open. Here at NIH, it's open source, non-commercial. So we take our tissues, we we coat slides with an adhesive that allows us to do the multi-parameter, multi-cycle imaging. We can do 6 to 8 fluorophores, fluorescently conjugated antibodies per cycle. We image on a variety of imaging platforms. We can then remove the cover slip, leave, do inactivation, and then rely on a repeated marker for image alignment. And so over the last few years, I've really led efforts to characterize a lot of different tissues in mouse and humans, and I'm grateful for the fantastic post-baccalaureate students that have worked with me as well, and I'll highlight their contributions to the project. And so in an interest of time, I just want to give you a flavor, just showing you the images, the kinds of image quality that we can obtain, the various tissues that we're looking at. And so here, this is just human skin. It's a challenging tissue because of autofluorescence, because of tissue lifting through fluidic cycles. And here I just show you 4 of 19 markers. I won't go through all the exhaustive markers except for a couple of details. I just want to show you that we can characterize diverse tissues. And so additionally, here's a liver, and I commented about the autofluorescence. So now the liver, for anybody who's imaged it, it's notably autofluorescent because of limits of lipids and other factors. But again, because of our optimal tissue workflows, we can get really high quality immunolabeling. And then additionally, with the spleen. So here's a human spleen. We see, the white pulp here, and then the fantastic open-ended capillaries of the red pulp. So we can really get after biology, and that confocal gives us a kind of a 3-dimensional quality. And again, this tissue, for anybody who's done the spleen, knows that it's highly autofluorescent because of red blood cells, but because of our optimal tissue workflows, we've overcome those obstacles to get very quality staining. And then finally, we talk about the thymus. So this is an image acquired by Rebecca Buchel. Again, so she's an undergraduate student. She's come to join the lab for 2 years and is now off to Harvard for her PhD. This is just 4 of 48 parameters of the lymph node that of the thymus that I've pseudo-colored to really just in 4 markers to highlight the diversity of the cortical epithelial cells. And it looks like a rainbow, but again, it's just 4 markers, and the differential expression of those 4 markers gives you this just hyperspectral looking thing. And so it's part of a collaboration with another consortium with the Germain, Teichmann, Abrasom, and Tegan Laboratory. And so then I really like this cartoon that comes from the ASCT&B working group with Kati Borner as a lead author, because it really nicely summarizes all the different consortium and the different tissues. And I just want to use it to highlight that, we have 7 human organs that we're characterizing, more and more developing. The limiting factor is access to tissue. We get it from surgical resections. We're also, complementary mouse tissues. And the marker number is just provided as a proof of principle because we've really pushed the lymph node to 65 parameters, the thymus to 48, and so on. And so then, of course, we want to scale and we want people to adopt this method because we really believe in what we created. In order to do that, we need to bring it to scale. We need to automate it. And so over the last year in the pandemic, that is exactly what we've worked on. And so these methods, in particular, here we have the methods, the aspects of the method, including immunolabeling and fluorophore activation. Now we've automated with a low-cost fluidics device. And then we've also removed the need for taking the sample on and off the stage, which could have problems with downstream analyses. And now we implement this available, all this kit is available for mortar instruments, these open chambers. And in total, we can acquire 20-plex in a really reasonable amount of time. The total time is incredibly affordable. And then we can do primary and secondaries along with FFPE and fixed frozen. And so I'll just show you, this is the overall workflow. Again, we're writing a very detailed protocol summarizing this because we want wide adoption. And so here we've taken this microfluidic device. The thing, the rainbow colored indicates a different panel or cycle number. It's pushed through into the imaging chip, and we put the tissue on a cover slip and create this closed chamber that accommodates 300 microliters of antibody. So it's very cost effective when it comes to antibody labeling. And then all the antibody labeling and dye inactivation has happened automatically. And a special credit to Colin Chu, who's a visiting fellow who was instrumental. We worked together very hard to get the automation, along with James Marr from Leica. And so now I want to show you the quality of data that we can generate using this method. And so here is a jejunum that I captured from human tissue samples. And I want to highlight all the efforts that we go to pin the tissue, to get it flat, to cut consistently across the villi so that we have consistent way of looking at the tissue. That's all captured here. And now I will cycle through the different parameters and you can just look at the quality. And so here's just the different markers. It's pretty boring. And then we'll zoom in so you can see, I just want to show you, these are the tissue markers. We can push it up to even more than 6 cycles. And then when we zoom in, because I always love to, as a microscopist, I always want to look at the data, play with the raw data. You can see the membrane stain, the CD49 is a mesenchymal marker along with Vimentin stained stroma. And we have really nice, it's confocal-like, but it comes off a wide field instrument. And so then we can use that for downstream analyses. And so then importantly, we want to make sure that we can also not only scale it and that it's automated, but scale it in terms of the volume of tissue, or really the area of tissue that we can acquire. And this is a human FFPE kidney section that I acquired that I'm showing on the automated method. I'm only showing 4 of 16 parameters, but you can see the scale bar. So you can realize that we can capture big tissues. And then on the left, because I always like numbers, is the standard image acquisition properties. These are not limited, but this is generally how we acquire the images with our 40x objective. This is the pixel size in the manual or automated, the dynamic range, and the total time of acquisition for rough estimates of a 24-plex. And then, so again, it's iterative. So we rely on, both the manual and automated methods depicted here. We capture multiple different images or multiple different cycles. So we need to align them so that we can do the downstream analyses that exactly like Monica described. And so I really credit some very fantastic computational scientists at the NIH, emphasis again on open source and freeware. And so they developed these platforms, and this is a schematic depicting the registration. And there are many different registration platforms out there. This is one that we found that works well for us. So you have these, the individual imaging panels. We have a repeated marker that, a marker that's repeated throughout the different imaging cycles that we can use to align. And nice thing about this with the versatility of IBEX is that we can use a range of markers for alignment. We've tested it on nuclear and membrane and structural. After alignment, we get quantitative metrics of how the performance, how it's performed. And then I've shown, because I can capture and align up to 260 gigabytes of data, and we could even push that further. And importantly, for someone like me, there's no programming experience required. And then we can execute this as an Amaris extension. And Amaris is a common imaging analysis software, and now they offer a freeware version for visualization. So, and that's all included in the protocols that is under review. And then, so we've acquired these images with multiple different cycles. We want to make sure that the images that we acquire are suitable for answering a biological question and then downstream analyses. And so one of the projects that I've worked on is looking at the normal and tumor lymph node, focusing with colleagues from the NCI on follicular lymphoma. And so we know that the tumor B cells have intimate interactions with a diversity of cells present in the tumor microenvironment, including myeloid cells and exquisite stromal cells. On the left here is all the protein biomarkers that I'm targeting with antibodies designed with IBEX panels. And so now I'm just going to walk you through the individual zoomed in follicles to show you why imaging is so important. And of course, this is a spatial crowd, so we appreciate that already, but that you can have similar cellular compositions, but then their distribution and patterns emerging from the tissue are very different. So on the left is a follicle from a normal lymph node. On the right is a follicle from a follicular lymphoma. And already you see there's incredible disorganization. We've lost our germinal center, we've lost our beautiful mantle zone. And then I'm just going to go through each of the markers here so you can see, the cellular resolution we're able to obtain. And it's shown as a 8-micron Z-stack because it's acquired with a confocal. So, just seeing the different spatial patterning and the kind of markers that we can look at in situ. And so we get nuclear markers, the differential expression, you can see that even with the dynamic range is only 8-bit, and so on. So I'll just stop talking as you go through the markers. Again, these are some of my favorite, the follicular dendritic cells, a challenge to segment them and treat them as individual cells indeed. And so on, and a different stromal marker, the loss of these fibroblastic reticular cells in the tumor, along with this really nice patterning. And with the ASCT, we know that there's naive B cells forming this little mantle region, and it's completely lost, and so on. So again, a challenge to the group would be understanding how we can interrogate patterns and analyze patterns across tissues. And so, yeah, so going quickly through the individual markers, so you see the staining pattern and their distribution. And then finally, and we've partnered with data scientists at Boston Gene to analyze the images that are coming from the IBEX method. Many of the strategies they're employing are ones that are developed by the pioneers here on the call and in the consortia. So I won't go into great detail other than say we can take the 39 markers that I showed you. We can do object-based segmentation using a nuclear marker and CD45. Then we arrive at these different populations based on their mean marker expression, which we can then, for all the patients, project into this HuBMAP space. And so we see like the tumor B cells are really nicely segregated over here. There's a diversity of populations that exist there. And then we have, for some cells, we can get some endothelial, we can get some lymphatics, but we're treating them, the stromal cell populations, in a very different way, actually using the pixel-based approaches that developed in the laboratory by Nishant, but also Michelangelo's group. We have, unlike single cell, we have really nice separation between 8s and 4s and a diversity of TFH cells that live here along with your Tregs. And so, again, just demonstrating that IBEX is definitely compatible with these downstream image analysis, that we can quantify them across the samples and make comparisons. This is just some of the analyses we're doing. We can look at the heterogeneity of the tumor B cells and normal B cells in a wide variety of ways. Here's just one summary of it. And then again, looking at the T cells and their diversity, and coming across actually, realizing unique populations that appear in some of the samples. And then it comes up in the analysis and we can, go into the image and see that that population does indeed exist. And so similar to Monica, yes, indeed, we can take these segmented cells, we can pseudo-color them based on their phenotype and put them back into, because we have the spatial coordinates, we can put them back in their native tissue context. And here's a normal lymph node, and we can see 3 different secondary follicles with germinal centers shown here, here, and already you see a diversity just in germinal centers, just in normal tissues. And then again, expanding on the work by many of the pioneers on HuBMAP, these community-based analyses. So implementing those workflows where we know that it's not enough just to have the cells, we want to find their neighbors. And I think it's really wild because you see unique little pockets of green, which corresponds to an enrichment for certain populations of T cells. We see a really nice germinal center in black and different areas of anatomical structures and niches here. And then, yeah, that's it. I appreciate it. I was going perhaps fast. I wanted to give time for questions, but I just want to close by thanking Ron. He's been fantastic. Again, I started with my postdoc and continued on as a staff scientist. I want to thank Rebecca Buchel. She's been fantastic in developing, a lot of the panels and characterizing, and many of you have worked with her on some of the HuBMAP work. So that she keeps saying fantastic, but she is, and she's off to Harvard to start her PhD program. Also an acknowledged past students who've helped me and then gone on to their MD and PhD. And then of course, my colleagues with the computational support, Colin and James and many others for helping with IBEX. And then my NCI colleagues for the tumor samples and follicular lymphoma, as well as supplying other samples that we've used to benchmark this technology and methods, and our Boston Gene colleagues. And then I want to close by putting a plug, if you're passionate about multiplex imaging and tissue-specific imaging panels, please join Jeannie and Neil on Wednesday to discuss these things. And I'll turn the camera on and take questions.
All right, thank you so much for that presentation. There's a couple questions in the chat. Earlier, someone had asked if your liver imaging protocol was available with details and how you overcome the autofluorescence problem in the liver.
Yeah, fantastic. So that's exactly what we want. So indeed it is. And like I said, we wrote a very detailed nature protocols because the effort is, hey, we want to help you. We want to support you. So please, you have my email, I think, please email, and I'm happy to share and happy to contribute. And really, I mean, we can only do one thing, we can't do everything. So we want to support HuBMAP And that's exactly what I was hoping would come out of this talk. So please, yeah, bother me. It won't be a bother.
Awesome. And then Fiona asked, how do you label your antibodies?
Oh, really great question. And they're all commercially available, so they come fluorescently conjugated. And for the fixed frozen, many times we can employ the direct conjugate. And then if need be, we can do secondaries, but it's limiting as the IBEX doesn't destroy the epitopes. And we have done some in-house conjugation. So again, I'm happy to you can email me. I can tell you what I think about, my honest to goodness truth, what I think about the different conjugation kits available. And so, yeah, please ask questions.
And then someone asked if you did staining on frozen human liver tissue and if you could share the antibody panel for human liver tissue. Yeah,
so yes, the answer is, and again, that's included in the manuscript, which we hope as soon as we get, we'll put it on bioRxiv and share with other people, but exactly how to handle the tissue, the antibodies, the order, and so on. And we could definitely, yeah. So anyway, yes, answered your question. Yeah.
And then someone asked, have you considered combining protein and RNA markers?
Really great question. The answer is yes, always, and we haven't implemented it, but that's definitely, that's where it's at, the spatial transcriptomics and protein, for sure.
Awesome. And then can I just drop your email in the chat?
Please, yeah.
Okay. Great. All right. Well, thank you so much. And if anyone else has other questions, again, like feel free to put them in the chat. And then Andrea, if you want to just check in there, if other people have questions or anything, feel free to continue that discussion. And otherwise, we will jump over to our next presentation, which is Nils, and I'll let you take over.
Hey everyone, thank you. I think my presentation will be very much focused on the technical aspect of the science and technology seminar. So I'm going to talk a little bit about some updates in our visualization framework that we've developed for HuBMAP in order to show data in the HuBMAP data portal. And so I'm gonna go through a fairly large number of slides fairly quickly. This is really mostly an update, and I hope that some of you out there who are working with imaging data will be able to use the new functionality that I'll talk about. And we're always keen to hear from people who have needs for data visualization. And I think this should be quite interesting to many of you. So I want to first of all begin by acknowledging the person whose work I'm presenting today. So this is Elon Gold, who's a software developer in my team, who's been adding a lot of new features to our Vitesse visualization framework. And so why is all of this work that we're doing important? And so if you just want to take a quick look here at this slide, it shows you an overview of the data that's available in the HuBMAP data portal. This is directly from the HuBMAP portal homepage, and you can see in this chart here on the y-axis, you see all the different assays that we have in HuBMAP, and then the bars correspond to the number of data sets that are available, and the bars are colored by the organ types. And the striking part here is the number of assays that we're dealing with in HuBMAP, which means we have to support an awful lot of different types of data analyses, file formats, and ultimately also visualizations. And so this is why we developed this interactive visualization framework called Vitesse, which is designed to be integrative and so to support multimodal single cell and tissue data. So to build this framework, we had to create a bit of a tool ecosystem because there was not a lot of available infrastructure that we could build on for really full-blown web-based visualization, in particular visual exploration tools. So it all started out with, or all starts out with VIV, which is an image visualization library for high resolution, high bit depth, multiplexed image data that runs directly in a web browser and doesn't require any other server than a web server. Avivator is a tool that is built around VIV sort of as a demo to show the features of this library. And while it is mostly a demo, it can also be used to explore imaging data very conveniently directly in your web browser. all you need to do is to point it to a TIFF file or some other file, as long as it's in one of the supported formats, and you can start playing around with it. We also have Visar, which is a similar tool. Visar is more focused on next generation image formats stored in ZAR containers, which can be, for example, as you can see here, embedded in Jupyter notebooks. If you're writing Python code and you want to interact with imaging data very conveniently, you can do this with Visar. We then created Vitesse around Viv, and so Vitesse is the tool that I'll spend most of my time talking about. So this is this visual integration tool for the exploration of spatial single cell experiments. That's actually an acronym. Here's just a little example of how this can be used to look at Visium data from 10x. And you'll see there are many other examples of how Vitesse can be configured to display all sorts of data that HuBMAP is generating. HuBMAP, the HuBMAP data portal includes a Vitesse instance that is directly embedded. So here's an example for Codex. You can see it supports sort of many channels. We can associate heat maps to show quantifications. We can show clusterings and many other types of information in these visualizations. There's also the Vitesse website where you, in this case, see a completely different example with a different configuration of views. But the most important thing here is that you can really load matrices, you can load embeddings, you can load images, you can, in this case, even look at individual molecules. This is an OSM fish data set. You can look at cluster level sort of quantifications in this view down here, where you see the expression by cell set and so on and so forth. To make it easier for people to create Vitesse-based visualizations, we've created a Python package. Here, another example embedded in Jupyter Lab. You can see in this example, we can also show genomic visualization. So these are metaxic profiles associated with the clusters that you can see in this HuBMAP scatter plot. And of course, we also have an R package, which is a bit less developed than the Python package, but you can see we can also create these visualizations directly out of R. So for example, if you have your single cell data as Seurat objects, you can embed these visualizations, in this case, directly in a RStudio instance, but you can also embed this in Shiny apps or in R Markdown. So that's the tool ecosystem. The data formats that we support is also fairly rich. So we can do OMI TIFF, we can something called OMI compatible ZAR for imaging data, as well as the next generation file format that's currently under development or has recently been sort of released by the OMI community. And they're all supported. In particular, these ZAR-based formats have the advantage that they are pre-chunked and can be organized in a way that makes it really convenient to retrieve data remotely and render them in a web browser. Vitesse supports also AND data, as long as it's stored in a ZAR container, so we can retrieve subsets of these large matrices that can be stored in an AND data file, as well as other annotations directly from Vitesse, directly in the web browser. All you need to do is put your AND data file or your ZAR store that contains your AND data information on a web server, and we can retrieve data from there. We can show genomic profiles, as I pointed out. We're using ZAR for that as well at the moment. All the image formats can be used in Vitesse, and then we have various JSON-based formats for image segmentation masks and other information. And so I want to spend a moment talking about the image segmentation support that we have. And so this is really where we have a couple of new things that I think fit in nicely with what we've heard so far. So the data formats that we support right now, so the traditional format was our own Vitesse JSON format that we used for both localization as well as cell shapes and other information to render cell masks based on, for example, cytokit segmentations. So we have that, that's already deployed in the HuBMAP data portal, and anyone can use it. Very recently, we added OMI TIFF bitmask as another format that we support. And the reason for that is that we also wanted to include support for larger images with many more cells than the custom format could support. And what we have already implemented and what will be coming soon is the support for the QPath segmentation format that is based on something called GeoJSON. And you'll see in a moment how that works. And so what is important to point out, and many of you who are familiar with image software are probably know how this works. So you can, in this case, associate cells or objects, I should say. This could be cells, but could also be functional tissue units or any other sort of thing that you want to segment out to various groups. So you can assign clusters, you can assign cell types, and many other things. So the test is very flexible when it comes to that. You can also link these objects to quantifications, matrices, for example, an N data matrix, if you have gene expression levels that you want to overlay on your segmented cells. And then VITESSE also allows you to select these cells, interact with them, create groups of cells, etc. And so what does this look like? So here is image segmentation support for functional tissue units. And this is based on the QPath format. This was created manually for the Kaggle competition. So here are glomeruli annotated and highlighted in yellow. And so I think for anyone who's played around with microscopy software as tools like QPath, etc., this is not super exciting, but we can do this. And this is all going to be available directly through the HuBMAP portal for our data sets. So we're looking forward to seeing more and more segmentations that we can make available directly through VITESSE in the HuBMAP data portal. So here's an example of what happens when you hover on a segmented object. And the other new thing is the OME-TIFF or the OME-TIFF bitmask. So here's a CytoKit-based segmentation that TCCMU provided to us as a sort of a demo data set to work with to ensure that our segmentation visualization works. So the example that you see here only has 3 channels activated in the image. And you see the data set overall has 4 antigens. And you can obviously zoom in and then we can overlay the cells. And what you see here, which is all new, we can now not only show like cell segmentations, but also subsets or more detailed structures. So we can do cells, we can do nuclei on top of it. We can look at nuclei boundaries separately. We can look at cell boundaries as well. And then we can overlay, for example, if we go a little further, here's the DAPI channel overlaid on top, the quantifications derived from the DAPI channel overlaid on these cell segmentations. Here's the CD45 channel overlaid. And all of these things are interactive. So you can hover on any cell, you can see versus expression profile in the matrix. And then, as I said, we can also combine this, oops, that was a little fast, with interactions. So again, you hover just on these cells, you can see all the cluster assignments, you can change these color mappings on the fly. And as I said, this could come out of an ANDATA file, for example, or from some other file, and we can play around with this. It's very fast, and all you need is a web browser to do this. So there's no need for any other software. And I guess what is going to get very exciting is once we're moving into 3D. So we've just added support for 3D raycasting in our Viv image rendering library. So this will come to the tests very, very soon, and will also be available in the HuBMAP data portal. So we're looking at a 3D IMC data set here. Again, all you need is a web browser. The data is living in an OME-TIFF file in the moment. And you can add channels, you can do color mapping on the fly. But the next step here clearly is to bring segmentations also in these 3D data sets. And this is something that we're very excited about, and looking forward to working with members of the community to figure out how we can support this. And if anyone is interested in trying to use VITESSE in their own work, as I pointed out earlier, it is supported, for example, directly through Python in Jupyter Notebooks or Jupyter Lab. If you're working with that, you can build these visualizations yourself. We have a lot of a lot of demos on our website. There is also documentation for tool developers. So if you're building your own web-based tools, you can use our JavaScript implementation. Or if you're more like a data analyst and you're writing code to analyze data sets, you can use our Python or R package, which will then generate a JavaScript-based visualization and embed it in your data analysis environment. So that's it for me. I think just on time, just about time to wrap up here. So if there are any questions, I'm happy to take those either now or via email. Or via Twitter. I guess I don't have my email address on here. Thank you.
Thank you, Nils. Yeah, I don't see any questions in the chat, but obviously if anyone does have questions, you can send them over. And with that, I think we're all set. Thank you everyone for coming today. Thank you to our presenters for great presentations and to everyone who engaged in the chat and discussion. And with that, we will hopefully see you all at our next event.